Good morning, and welcome to the course. My name is Wesley Martin, and this is Education Lesson. Over the next several weeks, we're going to study physiology. And the simplest way to define that word is the study of how the living body works. Anatomy tells you what the parts are. Physiology tells you what they do. My goal today isn't to give you details to memorize, it's to hand you the four big ideas that the whole rest of the term hangs on. If you walk out remembering nothing else, remember these four. Function follows structure. The body fights to stay balanced. It does that through feedback, and all of it happens at the level of the cell. Everything we cover later, the heart, the kidney, the nervous system, is just those four ideas playing out in different places. So let's start with the first question. What is physiology? Physiology is the study of function, how living things work, from a single ion channel all the way up to the whole organism. And whenever a physiologist looks at a process, they're really asking two questions at once. The first is mechanism. How does this actually happen? What are the physical and chemical steps? The second is purpose. Why does it matter? How does this process help the organism survive and adapt to its world? Hold on to both of those questions because they're the lens we'll use all term. When we study how a muscle contracts, we'll ask both how the machinery works and what problem contraction solves for the body. Good physiology always connects the two. A mechanism with no purpose is trivia, and a purpose with no mechanism is just hand-waving. We want both. You'll often hear these two words together, and there's a reason. Anatomy is structure, what the body is made of, and how its parts are arranged. The shape of a bone, the branching of blood vessels, the layout of a cell. Physiology is function, what those parts actually do and how they do it. How that muscle generates force, how that kidney filters blood. Now, here's the key idea, and it has a name: complementary. Structure and function always explain each other. Complementarity. Structure and function always explain each other. The shape of a thing makes its job possible, and you can't fully understand one without the other. A blood vessel, a blood cell, is a flattened disc because that shape maximizes surface area for carrying oxygen. So all term, whenever you meet a structure, ask what it's for. And whenever you meet a function, ask what its structure makes it possible. The body is organized as a hierarchy, and it's worth seeing the whole ladder at once. At the bottom is the chemical level, atoms and molecules they build. Those molecules assemble into cells, which are the smallest units that are actually alive. Cells of the same kind group into tissues. Different tissue combines into organs, like the heart or the stomach. Organs that work together form organ systems. And all the systems together make up the organism, you. The important point isn't the list, it's that each level emerges from one below it and depends on it. A property like pumping blood doesn't exist at the level of a single molecule. It appears only when you assemble the right structures. So physiology asks how function arises at every rung of this ladder, and how the rungs connect. Here are the 11 organ systems that share the work of keeping you alive. Don't try to absorb all of them now, we'll spend real time on most of them later. But notice the variety of jobs. The integumentary system, your skin, is the barrier. Skeletal and muscular give you support and movement. Nervous and endocrine are the two control systems. We'll come back to those a lot. Cardiovascular and lymphatic move things around and defend you. Respiratory handles gas exchange. Digestion handles fuel. Urinary handles water and waste. And reproductive carries the species forward. The single most important thing on this slide is the box in the corner. None of these work alone. They cooperate constantly. When you exercise or get sick or even just stand up. Several of these systems coordinate in real time, and that cooperation is most of what physiology studies. This is the heart of the whole course, so I want to slow down. Homeostasis is the maintenance of a stable internal environment in the face of constantly changing outside world. Think about what your body pulls off every day. It's freezing outside, or it's 100 degrees. You've eaten, or you're starving. You're sprinting, or you're asleep. And yet your core temperature, your blood sugar, your blood pH barely move. That stability is not an accident, and it's not free. Your body is working continuously to defend it. Almost every system we study exists, at least in part, to serve homeostasis. So as we go through the term, keep asking the same question. What variable is this process protecting? And what would happen if it failed? That question will organize everything that comes next. Here's a subtle but important point. Your cells never actually touch the outside world. They're bathed in fluid, extracellular fluid. And that fluid is what homeostasis really works to protect. We divide the body's water into two compartments. Extracellular fluid sits outside the cells. That's the plasma in your blood, and interstitial fluid in your tissues. Intracellular fluid is the cytosol inside the cells. Now, the composition of these two is strikingly different. For example, there's far more sodium outside cells and far more potassium inside. The cell spends energy to keep it that way. And as we'll see, those differences are exactly what makes nerves fire and muscles contract. So when we say the body defends the internal environment, what we literally mean is keeping that extracellular fluid, the sea your cells live in, within narrow limits. So what exactly does the body keep constant? Here are some of the big regulated variables, each held around a target value called a set point. Core temperature sits near 37 degrees Celsius. Enzymes only work well in a narrow band. Blood glucose stays roughly 70 to 110 milligrams per deciliter. Because it's your cell fuel, blood pH is held between 7.35 and 7.45. Proteins literally change shape if it drifts. Blood pressure runs around 120/80 to keep tissues supplied. The plasma, calcium, stays between about 8.5 and 10.5, because nerves and muscles depend on it. I genuinely encourage you to learn these numbers. They'll come up all term. But the bigger idea is this. For each one, when the value drifts away from its set point, a control system notices and pulls it back, which raises the obvious question, how? And the answer, most of the time, is negative feedback. It's the body's default control loop, so let's walk the circuit. First, a stimulus. Some variable leaves its normal range. A receptor, a sensor, detects the change. It sends that information to a control center, which compares the value to the set point. The control center then activates an effector, which acts to oppose the change and push the variable back. The word negative just means the response counteracts the disturbance. Take body temperature. You overheat. That's the stimulus. Temperature sensors in your skin fire. Your hypothalamus, the control center, registers that you're above set point. It triggers your sweat glands, the effector, and evaporation cools you back toward 37 degrees. Then the loop quiets down. Notice it's self-correcting and self-limiting. Once you're back to normal, the signal fades. That's the pattern behind most regulation in your body. Now the mirror image, and it surprises people. In positive feedback, the response amplifies the change instead of opposing it. It pushes a process faster and faster toward completion. That sounds dangerous, and uncontrolled it would be. So the body uses it rarely and only when there's a clear endpoint that shuts it off. Two classic examples. Childbirth. The baby stretches the cervix, which releases oxytocin, which strengthens contractions, which cause more stretch, which releases more oxytocin. The cycle escalates until delivery ends it. Blood clotting. Activated platelets recruit more platelets, so the clot builds rapidly until the break is sealed and the signal stops. So here's the contrast to lock in. Negative feedback keeps things steady. Positive feedback drives them to an end. Same logic of loops, opposite purpose. Most of the body runs on the first. A few critical events need the second. We've been talking at the level of the whole body. Now we're going to zoom all the way in. Because all of this, every set point, every feedback loop, ultimately happens inside and across cells. The cell is the smallest unit that actually carries out life. And the thing that makes a cell is its membrane. The thin boundary that separates inside from outside and decides what's allowed to cross. That membrane is the reason the internal environment can be different from the external one in the first place. For the next few slides, we'll look at how it's built, how things move through it, and why the gradients it maintains turn out to power some of the most important processes in the body. So let's start with the membrane itself. The plasma membrane looks simple and does an enormous amount. Its foundation is a phospholipid bilayer, a double sheet of molecules with water-loving heads facing out toward the watery fluid on both sides, and water-fearing tails tucked inward, away from water. That arrangement forms spontaneously and gives you a stable barrier. But a pure barrier would be useless, so the membrane is studded with proteins, channels, pumps, and receptors, and those proteins do most of the real work, moving specific substances and receiving signals. The result is a membrane that's selectively permeable. Some things slip across freely, others need a protein to help them, and others are blocked entirely. That selectivity is the whole point. It's what lets a cell hold an internal composition completely different from its surroundings, and, as we'll see next, what lets it move things deliberately rather than just letting them leak. So how do things actually cross? Two broad categories, and the dividing line is energy. Passive transport requires no energy from the cell. Substances simply move down their gradient, from where they're crowded to where they're sparse. That includes simple diffusion, osmosis, which is just the diffusion of water, and facilitated diffusion, where a protein provides a path, but the molecule still flows downhill. Active transport is the opposite. It costs ATP because it moves things against their gradient, uphill. The classic example is the sodium-potassium pump, which constantly pushes sodium out and potassium in to maintain those differences we talked about earlier. There's also secondary transport that borrows one gradient to power another, and bulk transport, endocytosis and exocytosis for large cargo. The takeaway: if it goes downhill, it's usually free. If it goes uphill, the cell has to pay for it. I want to pull out a principle here, because it's one of the most reusable ideas in all of physiology. Things flow down gradients, and the body spends energy to build gradients, precisely so it can put their flow to work later. It's like pumping water uphill into a reservoir, so you can run it back down through a turbine. Watch how often this shows up. Nerve impulses. The cell maintains ion gradients across its membrane, then lets them flow to generate electrical signals. Gas exchange. Oxygen and carbon dioxide diffuse across the lungs simply by following partial pressure gradients. Kidney filtration. Pressure and solute gradients shape what gets filtered and reabsorbed. Same idea every time. Store potential energy in a gradient. Release it to do something useful. Once you start seeing gradients, you'll spot them everywhere. This term. And a lot of physiology suddenly clicks. We've built up cells, membranes, gradients, but a pile of cells isn't a body. Trillions of cells only behave as one coordinated organism because they're constantly communicating. So the last theme today is communication and control. How cells and systems send and receive signals to act together. This is what makes integration possible. It's how your heart knows to speed up when your muscles need more blood. How your pancreas knows to release insulin after you eat. There are two main signaling languages in the body, chemical and electrical, and two master systems that coordinate everything, the nervous and the endocrine. Let's look first at the chemical messenger cells used to talk to each other, and then at how those two big control systems divide the work. Cells talk to each other mostly with chemical messengers, and a clean way to organize them is by how far the signal has to travel. In autocrine signaling, a cell releases a messenger that acts back on its own receptors. It's talking to itself, often to regulate its own activity. In paracrine signaling, the message acts on neighboring cells in the same local tissue, short range. Synaptic signaling is specialized, very short range case. A neuron releases neurotransmitters across a tiny gap, a synapse, onto a specific target cell. And endocrine signaling is the long-distance option. Hormones are released into the bloodstream and carried all over the body to reach distant targets. Same basic idea throughout. A messenger and a receptor that recognizes it. But the range changes everything about speed, specificity, and how the signal is used. That distance axis sets up the next slide perfectly. Here are two systems that coordinate the entire body, side by side, and they're beautifully complementary. The nervous system signals with electrical impulses and neurotransmitters. The endocrine system signals with hormones carried in the blood. Nervous signaling is blazingly fast, milliseconds, while hormones take seconds to hours. But there's a trade-off. Nervous effects are brief, switching on and off quickly, whereas hormonal effects are prolonged and lingering, and they differ in targeting. The nervous system is like a phone call, a specific message to specific cells. The endocrine system is more like a radio broadcast. A hormone goes everywhere, but only cells with the matching receptor respond. Neither is better. Fast and precise is perfect for yanking your hand off a stove. Slow and sustained is perfect for growth, metabolism, and the stress response. Between them, the two systems cover every timescale the body needs. Let me tie the whole hour together with one ordinary example, or in our case, 20 minutes. You go for a run, watch how many systems jump in at once, all serving that one goal of homeostasis. Your muscular system starts demanding far more fuel and oxygen. Your respiratory system answers. Breathing deepens and quickens to pull in more oxygen and blow off carbon dioxide. Your cardiovascular system raises heart rate to deliver that oxygen-rich blood where it's needed. Your nervous system is orchestrating all of it in real time. Your endocrine system releases epinephrine to mobilize energy stores. And your integumentary system, your skin, starts sweating to dump the extra heat your muscles are producing. One simple act, and nearly every system is cooperating to keep your internal environment steady. That is physiology. Not a collection of separate parts, but an integrated whole. So let's close where we started. The 4 key ideas to carry out of this room. 1. Structure and function explain each other. Anatomy and physiology aren't two subjects, they're one. 2. Homeostasis is the goal. A stable internal environment is what keeps your cells alive, and most of what the body does is in service of it. 3. Feedback is the mechanism. Negative loops hold variables steady. Positive loops drive a few critical processes to completion. And 4. The cell is where it all happens. Membranes, gradients, and signals are the machinery underneath every system. Hold those 4 ideas, and the rest of this term will feel less like memorizing facts and more like watching the same logic play out in new places. That's the goal. Next time, we'll take the first of those systems and dig in. Thanks everyone. Be sure to subscribe for more education lessons.